would like to go to the panel, and I think, Wendy, are you still on the?
Yep.
Okay, great. So I think we'll open up the floor for questions.
Rick, that's absolutely fascinating and obviously important. So going back to a question I asked Skip earlier, I think you were in transit, in which viruses did not seem to have the same window of opportunity early in life to impact immune function. In a germ-free animal, I mean, in an SPF animal, I can see with weaning, there's a profound change in the gut microbiota. We know more about bacteria than other things, but in a germ-free setting, why do you think that same window of opportunity occurs? Is it an epigenetic factor? What is it? Food stimulated? What's the deal?
Your mic is not on.
If I could reframe Balfour's question, what's the deal? How's this happening? And why is this window so important? And for NKT cells, IgE, Treg cells in skin and the lung. There's presumably some way we're working on it right now in terms of our current hypothesis that there is a window of development, of immune development, that is very important for cells to establish their niche. So NKT cells are quite interesting because they're actually tissue resident cells. They come in early postnatally and expand through a brief window. What's regulating that expansion is really what's, I think, the critical question. And what are the cell types that are required for both that infiltration to the tissues and that expansion? And how are microbes then influencing that? And there are probably multiple different ways that microbes are doing it. It's interesting that we were just really quite lucky, I think, because of Dennis's interest in sphingolipids through Bacteroides, that we were able to bring up these ideas in the first instance, that Bacteroides happens to have at least one example of a mechanism to regulate that expansion. And as Ding Ding showed very nicely in her paper, that bifrag glycosphingolipid is able to inhibit that proliferation in early life. But I think it's a developmental process, Balfour. That understanding what those developmental events are during early life and what's regulating them and how the microbes are regulating them is really what the critical question in our mind and what's driving our research right now. It's interesting, there are other things that are regulated in early life that are not necessarily microbially regulated, they do regulate the immune system. So it was a very important paper, I'm blocking on the first author, but it was a Nature paper in 2013, I think, that showed that there's a group of erythrocytes that are very temporally regulated in mice during the first 2 weeks of life that are CD71 positive. And they're only present during those first 2 weeks of life and not thereafter. And they're very, very important in regulating the host's response to microbes. And if you remove those CD71 positive red blood cells, the host responds excessively, myeloid cells respond excessively to microbes. And so that's an example of a developmentally regulated immune event that is not microbially regulated, but shows that there are developmental events going on in the immune system. We know that there's development of the immune system. Look at gamma delta T cells, for example. They come out of the thymus in waves and go to the intestines and the various mucosal tissues at particular times of life, and they become resident in those tissues. I think, as it turns out in hindsight, NKT cells are just another example of a resident cell that are coming in a particular critical time of life that are then being regulated by processes that we're trying to unravel. But that's our current view of it and what we're trying to understand in my lab right now.
So a comment and a question. Firstly, I didn't say that viruses don't have these effects early in life. What I should have said that I wasn't smart enough to have done the Blumberg experiment to try it in 2-week-old. In adult mice, the viruses can have very dramatic things, but an interesting thing by way of comment is that the types of immune responses that we showed are altered by chronic virus infection are very different than what Rick is describing. They're much more interferon gamma, Th1 regulated kind of responses, so there could be a difference there. So question to both Rick and Wendy. Thank you very much for your beautiful talks. So the whole summary of all of it, including the cancer part in this presentation, is that the mouse models are substantially altered by the presence or absence of different microbes. And I spoke about viruses. If we had Miriam Murad here, she would tell you about a protist. If we had people studying fungi, they would tell you about fungi. They would all say the same thing. So can you comment on how we can make mouse models more useful if in fact they're differentially colonized in all these different settings? Or do we just accept the diversity and work out the mechanisms? But these are huge effects that are being observed here in all of them. So I wonder if you would comment on that. Your mic is off.
Yeah. Wendy, you want to try to take it, or I can handle that. That's a very important question, but Wendy, you want to go first?
Sure. I think Andrew McPherson is going to speak tomorrow a little bit about standardization and what that means in model microbial communities. I think there's been fantastic work that we're going to hear about tomorrow from pet shop mice, if you will. And I think they all provide fantastic opportunities to discover and uncover biology. So if we hadn't just looked with our eyes, we would have underappreciated the contribution of tritrichomonads, protists, to the initiation of type 2 immunity. But sometimes when you want to set out a causality experiment, I think it's very, such as proving that fuso maybe potentiates colorectal cancers, or that iNKT cells are influenced by the microbiota in a critical window. Having a defined community, knowing what's there, whether it's a mono association or a mock community of 8 or 30, will be very important. So I think everyone's approach to the microbiota, whether it's pet shop or conventional or standardization, is valid. And we just need to be very open in what we're doing and why, hopefully, as well.
Also, it would also be really, I was not here for this morning, and I regret it, because the talks sounded wonderful. I was coming back, unfortunately, from another trip. But the question is, can we utilize some of these other models, whether it's C. elegans or Drosophila or whatever, to try to get at some of these questions? Because if it is developmentally regulated, like we hypothesize, you would expect that there would be developmental events that are age dependent that are occurring in these other organisms, some of which may have similarities to things that we're discussing, but maybe there are developmentally regulated events in these organisms that are unique to those organisms that also be amenable for study to try to understand the developmental regulation, how microbes affect that development. And it's also possible that even though a virus doesn't per se affect, let's say, have an age-dependent effect, it might have an effect on one of these other aforementioned pathways.
Yeah. So on the standardized thing, I'm just going to throw it out there. If you pick the wrong standard community, first of all, I believe that the standardized models are great, but you might have left Trichomonas mirus out. And so you have to be very, very careful about proscribing a single way to do these kind of experiments. Otherwise, you'd miss both of these things.
I mean, it was really, I think Wendy's example is a terrific example of just great observation. It's a perfect example of just using your eyes and really keeping your eyes wide open. Totally agree.
Okay, great. I want to make sure that we get the last two questions in, but we'll have to go kind of quickly to keep on time. So Gary.
Okay, thank you, Wendy and Rick, for your wonderful talks. Rick, as you know, the human microbiota early on in life goes through a fairly orderly succession, and your story about Bacteroides fragilis and the biochemistry with sphingolipids maybe establishes proof of concept that through the biochemistry of the organisms that are relatively simple early on in life, you might be able to understand when immune tolerance actually occurs in humans. So it's a little bit out of a box question, but I'm wondering if you could speculate on that.
It's a great question, Gary. Gary asked whether there could be, it's about establishing immune tolerance. And that's one of our hypotheses, what's going on with NKT cells. That it's a way to establish a type of NKT tolerance, so the NKT cells are not responding inappropriately to later life environmental triggers, but we don't understand the mechanisms of that. But I think you're right. Whether that's happening with T cells or other resident cells in those tissues, you wouldn't expect tolerance to occur necessarily to cells that are migrating through the tissues. Although I guess you could be having systemic tolerance and tolerizing them to specific events as they're earmarked for going to the tissues later. But for the resident cells that are there, I think, and that are going to be there for the rest of the life of the host or a long part of the life of the host, you really want to establish tolerance. So I think it's a very important question you're asking.
Yeah, I was wondering specifically, could you map back to the bacteria based on specific molecules like sphingolipids, exactly what type of bacterial composition would induce immune tolerance, just because the succession early on in life is fairly well defined in infants and human infants?
I hope that the Congress is listening right now because we need money. These are great questions for experiments, but we really need money. The community needs money to fund the investigators to do these kinds of things because they are really important for human health. And to try to take some of the information from human succession and bring it back to the mouse, as we heard from Jeff and many others during the course of this day. So these are very important things to do.
Okay, one last quick question.
Thank you. I really enjoyed both of your talks. And I'm curious about, and I appreciate the plug for C. elegans as well. But I'm really trying to draw the parallels between the two studies, because it seems to me that this, and whether or not there's any basis for it between your two groups or elsewhere, but it seems to me that these critical periods, there are mechanisms that open them and close them. And I'm wondering whether or not similar mechanisms can be occurring later in life to relate to the development of cancer and the like. And whether or not, and also thinking about the periods, these critical periods, and the types of microbes that are present during those periods, and whether or not they may specifically be influencing that process. And how would you go about the best way to making those parallels?
As I've hit into my 60th year, well past it, I'm sure that there must be other windows as well, and developmental events that are going on throughout life that are under the control of microbes. We're not studying that. I don't have a good answer for you how to study that. But for example, there's a paper that we have that's coming out really soon. It's a little different, but if you take ATG1601 deficient animals, we did this work with Arthur Kozer's group, and ATG1601 deficient mice will develop a spontaneous colitis if you age them. They don't develop colitis spontaneously under normal circumstances. Steph Targan's group has exactly the same data that's part of that same story that's coming out. So yes, there are things that are happening with aging in a genetic and environmental context that suggest that there are things that are just as important in later life as in early life. But I don't have a good answer for you how to study it, but you need a big budget again.
Okay, I think we, thank you very much, and I want to move on. Oh, Wendy, sorry.
Oh, I was just going to say, I think the worm and the fish models are fantastic for asking those aging questions about the microbe. So if that was Buck, I think you're going to be able to do some of that.
Thank you, Wendy.
Thank you. Okay.